also a representative of the DISCO Research Infrastructure, Distributed System for Scientific Collections, and I'm one of the two moderators for this BOF session today. The other moderator is Andrew Bentley from the Kansas University Biodiversity Institute and Natural History Museum in Lawrence, Kansas, USA. He also represents the Biodiversity Collections Network. And we're both very grateful for everybody turning up and participating in this session. And we're especially grateful for technical support being provided to this Zoom session by Matthias Dillon from the Meiser Botanic Garden in Belgium, and to help us Wouter Haddink from Naturalis in the Netherlands, and Gil Nilsson from the Florida Museum of Natural History, who will be helping to take notes of this session. So thank you all for joining us in this informal session of interested people. I have to inform you that the session will be recorded for later viewing, and I believe that the recording has been started. The purpose of the session is to explore the digital specimen and extended specimen concepts, finding out something about their commonalities and differences, and discussing how we can converge towards a common global specification. There will be some session notes in a Google Doc, and you can see the link there on the screen, bit.ly BOF01 notes. Perhaps somebody would be kind enough to post that into the chat, please. Please go there and add your name under the heading, the red heading, participants, to show that you participated in this session. Our session format today will feature two scene-setting presentations, each followed by a short question and answer session. First, I will introduce the concept of open digital specimens, and then Andy will introduce the extended specimens concept. We'll then open up the forum for a wider discussion, a free-form exchange of views for an hour or so, where topics may include any of the following, such as similarities and differences between digital specimens and extended specimens, other initiatives to bring into this discussion from specific projects and/or countries, development and implementation plans for digital specimens and extended specimens, the potential role of groups within the Research Data Alliance, such as the Biodiversity Integration Interest Group and the Data Fabric Interest Group, and identifying some next steps towards global convergence. And towards the end of the 2-hour session, we'll try to summarize and wrap up accordingly. Andy and I would like you to treat this birds of a feather session as an equal opportunity for all of us to contribute informally to explore the ideas. We ask you to be polite and kind to each other and inclusive, and to remember the TDWG Code of Conduct, which you can find on the TDWG conference website. The chat function will be available during this session for asking questions and making subsidiary points. Please use it carefully and appropriately. Improper use of the chat function may result in you being removed from the session or in the chat function being disabled. We would also ask you, please, to keep your microphones muted when you're not speaking. Please bear with us with any Please bear with us for any technical difficulties we may have, and we hope that you enjoy the event. Okay, so let's move to the two scene-setting presentations. These are two introductory presentations for orientation of about 15 minutes each or so. After each one, there will be an opportunity to ask questions for clarification. To do this, we ask you to enter your question in the chat. A moderator, either myself or Andy, will read out the question for the speaker to answer. During these question and answer sessions, please don't offer your comments or opinions at this stage. That will be possible later during the open discussion. So just questions for clarification. The structure of both presentations is roughly as follows. They start with a high-level description and characterization of where the respective concepts have come from. They describe something of the technical foundation and basis of each of the concepts, the main technical elements of a digital specimen or an extended specimen as appropriate, and the existing work that's in progress or planned, including implementation plans for extended specimen network and DISCO. Okay, so let's turn to the first of these presentations, and this is by myself to introduce the open digital specimen concept. Let's begin with a high-level description, characterization. Where does this idea of a digital specimen come from? On the one hand, it's grounded in needs. It's grounded in the needs of the DISCO research infrastructure in Europe, the distributed system of scientific collections, which is aiming to work towards digitally unifying all European natural science assets under common curation, access, policies and practices, and aims to ensure that the data relating to natural science assets is easily findable, accessible, interoperable, and reusable, fair. And of course, it's also grounded in the needs that can be derived by examples of work by, for example, Michael Webster and his colleagues in the extended specimen work that was published in 2017, and work by David Schindel and Joseph Cook in 2018 on trajectories for the next generation of natural history collections published in PLOS Biology. On the other hand, this digital specimen concept also comes from a technical angle. It has to be technically aligned to recommendations that are coming out of the Research Data Alliance. It has to implement the guiding principles of fair, findable, accessible, interoperable, and reusable. And from the European perspective, at least, it has to be compatible with the emerging federated environment that is the European Open Science Cloud that's gathering pace in Europe to deal with the storing, sharing, and reuse of research data across many, many different kinds of research disciplines. And fundamental to these recommendations, guiding principles, and compatibility with EOSC is the idea of a digital object that is represented by a bit sequence or bit sequences that can be stored in repositories, that is described by metadata so that it can be catalogued, indexed, and discovered, and is referenced by persistent identifiers so that it can be unambiguously identified. For specific kinds of digital objects, digital specimens provide an anchoring function for all kinds of data that can be derived from physical specimens. Data like genomic data, biochemical data, morphological data, geographical data, taxonomic information, species interaction data, ecological data, and more. The data that's locked up in physical specimens in natural history collections throughout the world can be released through digitization, through analysis, and through computational methods. As a kind of digital object acting on the internet as twins for physical specimens in natural science collections, digital specimens are more than just digital representations. They're actionable knowledge units on the internet that can be manipulated remotely across a network by machines and by humans. I want to stress that this aspect of manipulation by machines is often understated when we think about digital specimens and extended specimens, because traditionally in this domain of working with specimens, it's humans that work with physical specimens historically, and this idea of working with digital data is a relatively recent innovation, only in the last 20 years or 20, 30 years or so. As twins of physical specimens, digital specimens enable transformations of working practices in the future. For example, they permit wider access for research and learning through the packages of images and links to third-party data, and the ability to work with data where access to the physical specimen itself is not needed. Digital specimens allow us to attach annotations and interpretations, not just to a single specimen, but to all of the duplicates of that specimen that may exist in different collections. Digital specimens allow us to arrange loans and visits to the physical specimens, to attribute work done, the specimens that are used, persons credited with doing work on specimens, etc. Curation by the community becomes possible, and tracing the provenance of important specimens can be put into practice. We can insert relations between multiple specimens and between specimens and other data, growing a persistent identifier graph, a PID graph, that can be used for analysis and inference. In the same way that digital object identifiers, DOIs for journal articles, are growing a PID graph of journal articles and authors that allows analysis and inferences of the relationships between different aspects of science and research. Digital specimens can be organized and indexed into different virtual groupings of related specimens, for example, by gathering, as exicati, or in thematic criteria of your choice. And of course, digital specimens admit to machine processing, learning and analysis, and data mining on very, very large scales across all of the subdisciplines where that specimens provide the scientific basis for: taxonomy, phenology, biogeography, stratigraphy, mineralogy, and others. As such, these digital specimens on the internet bring transformation benefits that become embedded in the basis of value chains that originate in the natural science collections. From the initial gathering, through the science that's carried out based on specimens and the commercialization of the results of that science, through to the repatriation of the economic and the social benefits back to the points of origin of the original specimens, i.e. back into the countries from which specimens have been gathered. And you can see there across the screen a range of digital transformation benefits that range on the one hand from machine actionable and human readable, representing physical things in cyberspace with context and meaning, through the easy exchange between systems secured by design and trusted and part of open science clouds, to being easily citable by their persistent identifiers, and of scale, form and precision that is required for advanced data science, often unimaginable uses of this kind of data, which we can't foresee at the present time. Here's an simple example of a digital specimen in illustrated form. The top middle of the screen in the little blue box, you can see a physical specimen that has the catalogue number BMNH 2006 12 6 40 41. This is a physical specimen in the Natural History Museum in London. It is a paratype and it has a holotype of a corresponding specimen in the French Natural History Museum in Paris with the catalogue number JNC 1848 D1. This specimen has a number of relations and other information associated with it. We know it's a specimen of Halorchis castex, and we can tie that to the taxon concept in the catalogue of life. It has a representation in the Natural History Museum's data portal. It has a circumscription in Zootaxa and other publications. And over on the other side, you see that it has links to images, to a treatment in Pluzzy Treatment Bank, and to a sequence in GenBank. And there are many more examples of other kinds of specimens which have similar kinds of relations and data associated with it. Some is data specifically about the specimen, like what it is, where it was collected and who by. Others is data that is derived from it, like DNA sequences and CT scans and images and that kind of thing. And all of this data can be represented in specific formats such as JSON, JavaScript object notation. It can be packaged as a fragment or a unit of data and information, which can be exchanged between systems and processed. From this simple digital example, we can assume a generic model, a curated and authoritative open package of links to other data. A digital specimen is a container with a persistent identifier and a type definition of digital specimen. The persistent identifiers, the PIDs, within it are pointers that resolve to the locations of other items. The digital specimen itself, the physical specimen that is the twin of this digital specimen, high-resolution images of the physical specimen, label information, tissue samples, DNA sequences, and much more. The PIDs can have corresponding metadata embedded into the digital specimen to avoid the need to resolve the PID just to find out whether it's something interesting or not. Similarly, the PIDs can have corresponding content embedded in the digital specimen as well, such as low-resolution images to avoid the need to retrieve the high-resolution ones. A digital specimen can be an aggregation of other digital specimens as well. A digital specimen for a tray of insects can also include persistent identifiers pointing to the digital specimens for each of the individual insects in that tray. To convert this idea to reality, DISCO proposes the Open Digital Specimen Standard, OpenDS, to specify the structure, content, and format of a digital specimen. Just as ISO 32000 specifies the structure and format of PDF, portable document format, documents, but doesn't tell you what you can do with a PDF document, so too will OpenDS specify structure and content and format without limitation on what you can do. A specification for OpenDS would allow us, for example, to transfer digital specimen data from one system to another, to define operations to be performed remotely on one or more digital specimens in another system simultaneously, using a protocol such as Digital Object Interface Protocol. It would allow us to package one or more digital specimens for downloading from a system to a local system. And it can guide but not constrain the internal design of such systems, for example, by specifying the kinds of behaviors of systems in cases where systems are faced with unexpected forms of data. It has a technical basis, a data type definition according to the Research Data Alliance data type model that will build upon the concepts from ABCD3 with its EFG extension for geosciences and Darwin Core, taking into account the current alignment work that's been talked about and initiated over the last year to 18 months, most recently in the WS02 working group that took place earlier today. Digital specimens, OpenDS, have their roots in Foundry ontologies as well, extending where necessary the biological collections ontology, the ontology for biomedical investigations, the information artifact ontology. We can envisage, for example, a digitization event, which is presently undefined, as a new subclass of planned process in the OB ontology, alongside the specimen collection process, which is currently defined. And this would lead to a new class of digital specimen that would possibly be a new subclass of an information content entity within the information artifact ontology. And that would, of course, be the result of the digitization event. The OpenDS specification will also have to say something about how to serialize the object content to a specific format and representation, i.e. JSON, for exchange and processing purposes. So coming now to my last slide about the existing work that is in progress or planned, that has been taken largely in the process of designing and planning for the construction of the DISCO research infrastructure through a number of projects and initiatives. Initially in the ISTIG design study, we have developed a digital specimen architecture design and recommendations that has led us to a provisional data management plan for the DISCO research infrastructure. A year or so ago, we presented a vision for open digital specimens at the Biodiversity Next conference that took place in Leiden. And earlier this year, before COVID lockdowns, we were able to run a cost-mobilized OpenDS workshop in Warsaw, where the participants present agreed that there was a wide measure of interest in and acceptance of the idea of a digital specimen, even if not all of the details are yet understood and agreed. And some steps forward were identified, of which establishing liaisons more strongly between Europe and the USA, DISCO to IDIG-Bio and to the Biological Collections Network, and this birds of a feather session today to discuss moves towards a more global specification have been initiated. And the next event in this sequence of steps will be a plenary 16 discussion topic in the Research Data Alliance in November about starting a digital specimens or extended specimens working group within the Biodiversity Data Integration Interest Group of the RDA. So I think that's all I have to say about introducing open digital specimens. So I'm pleased now to take some questions.
I don't see any questions as yet in the chat. There's one here from Jarrett Pullen. How do you version digital specimens?
That's a very good question. The answer to that question is that when you resolve the identifier to a digital specimen, it will always recall for you the latest version of the digital specimen. If you choose to look up a citation of a specimen that has been made at an earlier point in time, there is a mechanism which will reconstruct the version of the digital specimen from its provenance information and change history for the moment at which it was time-stamped, i.e. when it was cited.
Anybody else? This one from David Shorthouse. Does a digital specimen require a centralized infrastructure?
That's also an interesting question. The answer to that is that it can be implemented in either a centralized or a decentralized manner. Both are possible.
And there's another one from Ian Engelbrecht. Do you possibly have an example to hand of a JSON representation of a digital specimen, even if just preliminary?
The answer to that is yes, we do. We will get back to you after this session to provide it.
Tim has just posted one in the chat there, I think.
Okay, thank you, Tim.
Another question from Donna D'Agosti. Is there a timeline when the ODS will be functional? Are there early adopters?
There is no timeline at the moment. The next immediate steps after this birds of a feather session are to begin to plan that timeline and to work out how the specification is going to be developed in more detail and who is going to participate in that. And we will be looking for volunteers to assist with that.
And then one from Mike Webster. Do you envisage the digital specimen concept to include digital data, e.g. media, collected during a particular collecting event, but not associated with a physical specimen?
It is possible to associate, I think perhaps this refers to what I believe Mike refers to as the difference between actual connections and conceptual connections. It's possible to make links between the specimen and data that has been directly derived from that specimen, i.e. an actual collection. And it is also possible to make links between the specimen and data which might be associated in some way with the specimen, such as information about its habitat or its behavior, or some sound or media recordings of another member of that taxon, for example.
Yeah, I think he may be referring to the difference between voucher specimens and observation records. So things that do not have a voucher specimen physically in a collection.
Yes, that is possible, yes.
Yeah. Then another one from
living specimens are also possible.
Another one from Jorrit Pullen. How would you implement a decentralized resolver? How does such a resolver stay in sync with their centralized counterparts?
We're proposing to use the handle system.
Okay, and one from Diane Rillinger. Do you anticipate connecting to primary literature?
Yes.
And then Deb Paul, any models for the community curation already imagined?
I don't have one in mind, but others may do.
Good set of questions.
Yes, okay. So I think if there is no more questions, we'll go on to the second presentation, which is from Andy, introducing the extended specimen concept. So I will stop sharing my screen and hand over to you, Andy.
Sounds good. Oops. Okay, good morning, everybody, or afternoon or evening, wherever you are in the world. So I'm going to be introducing the extended specimen and extended specimen network concepts that have been developed by the Beacon group. This originated, oops, there we go. This originated back in 2012 when a group of researchers and collections professionals came together, funded by NSF, to look at an implementation plan for what was then called the Network Integrated Biocollections Alliance. There were a number of goals that were outlined in that plan, one of which was to advance the engineering of the US biodiversity collections cyber infrastructure. There were a number of different points that were articulated below that, and one of those was anticipate the future of biodiversity specimen data integration. So then a little while ago, there was a 5-year NSF-funded research coordination network that was put together, an RCN called the Biodiversity Collections Network, that was funded to build a community to advance and sustain digitized biocollections. That culminated in a workshop that was held towards the end of 2018, which resulted in a publication called Extending US Biodiversity Collections to Address National Challenges. Within that report was this concept of the extended specimen that was obviously built out of Michael Webster's original concept of the extended specimen published in 2017. And there was a graphical representation of that extended specimen concept that was grounded in the physical specimen in the collection, and then all of these primary, secondary, and tertiary extensions to that specimen. The primary extension being the digital concept of that physical specimen housed in the collection, the digital record, any specimen media associated with it, and all of the metadata associated with the specimen. The secondary extension was all of those products that were created from that specimen, things like field notes and gene sequences and images from the field and morphometric data, isotope information. And then a tertiary extension beyond that, looking at ancillary products that somehow related to that physical specimen and its digital data. Phylogenies, traits, taxonomy, protection status, those kinds of concepts. Later on, this was then published in Bioscience in an article called The Extended Specimen Network, a Strategy to Enhance US Biodiversity Collections, Promote Research and Education. And within that was given another example of the extended specimen concept using Appalachian lichens. And here you can see the specimens collected in the field resulting in physical specimens in a collection, the digital representation of that specimen with a digital record and a specimen image associated with it. And then the secondary extensions of field images, tissue samples, morphological data, etc. And then all of those tertiary extensions of biotic integration, species descriptions, conservation status, etc. But once you start looking at this as an example from a true collection, so this is an example that I pulled from my collection, it starts becoming squirrely pretty quickly. So you'll see here that there is a specimen in my voucher collection that has a unique identifier associated with it of a particular fish. That is then related to a tissue sample that has a different unique identifier that is sitting in my tissue collection. That is then linked to the actual voucher specimen itself. There may be images associated with that particular lot of the actual specimens that the tissues relate to. There may also be X-rays of that particular specimen, as well as field notes associated with that specimen as well that have been digitized and associated with the record in the database. And then there are these ancillary products that are created through research ongoing from those particular specimens. So this is from a publication where there were images taken of the specimen, there were cleared and stained representations of that specimen, there were slides made of the morphology of that specimen. And so there's all these digital products that are being created through the research that's being undertaken on those particular specimens. The data in the collection is then obviously associated with additional products that are lodged with additional aggregators like MorphoSource with CT scan information. And this information is then represented on the web through either a local web instance where people can search your local collection, or going through IPT and being published out to all of the aggregators. And so my collection is available in FishNet, it's available in VertNet, it's available in GBIF, and it's available in IDIC-Bio. So multiple aggregators, all with the same information coming from that IPT instance. You then have a whole bunch of GenBank sequences that may be associated with the tissue contained in the collection, and all of those obviously have GenBank numbers associated with them. And all of those are then linked digitally through the publication in GenBank and also back to the original specimen using the link-out system within GenBank. You then have a number of publications that may be associated with that particular, with the particular research that's done on that specimen. In this case, it's two genetic sequence information papers, and that information may be represented in multiple different ways. So in the one, you actually have a table within the publication that links all those GenBank sequences to the actual voucher information. In the other, it's actually contained in supplementary information, which makes it a little more difficult for you to be able to make those linkages between the GenBank sequences in the publication and the actual information associated with the specimen. So the extended specimen concept, unlike the open digital specimen concept, is more of a concept than a technical specification. Our hope was that we would build on existing initiatives and infrastructure that's already out there, as well as identify the gaps that need to be filled in order to be able to link all of this information together. It is, however, grounded in the physical specimen, because the digital data is only one part of the extended specimen network plan, which we'll talk about in a little bit. But there is an understanding in the extended specimen concept that these digital objects obviously need to be fair. They need to be findable, they need to be accessible, they need to be interoperable, and they need to be reusable. Findable in terms of they need to have unique identifiers and robust metadata that define those particular objects. And they need to be indexed in order for them to be able to be unique and be found. They need to be accessible. Obviously, this information needs to be published, and it needs to be free and open in order for it to be able to be found and linked to other objects in the chain. It also needs to be interoperable. It needs to be resolvable in order for integration and attribution of collections to be done. And then it needs to be reusable. They need to be able to be replicated and combined in various different combinations, and they need to be licensed, and there needs to be provenance in order to be able to link all of these items together and find out where they come from. There is also obviously a necessary underlying digital architecture that is associated with this. There need to be unique identifiers that identify all of these individual objects, be they PIDs, be they GWIDs, whatever the case may be. And these not only need to be associated with the digital specimens, but also all of the digital products that are associated with those specimens, as well as the institutions and collections from which these objects come. You need to be able to group these identifiers together in order to be able to reuse them. So being able to group a whole bunch of digital specimens together in order to reuse them in a publication. They need to be annotatable. So you need to be able to annotate these items in order to be able to change them. And they need to be connected. You need connectivity between all of these identifiers in order to bring about integration and also attribution. So one of the things that we've been thinking about during this process is the concept of a blockchain-inspired network, where everything is transactional. So you have a transactional system of everything that's associated with a specimen is a transaction. You loan a specimen to somebody, that's a transaction on that specimen. You annotate that specimen and change its identification, that is just a transaction. And so this whole transactional system of being able to link all of these items together and be able to visualize all of the changes that are being made to those individual objects over time. This closely aligns with the open digital specimen concept as it has been portrayed by Alex. But as we know, there is a social component to this whole thing. Our world is very complicated and very complex, and extremely complicated in terms of the number of players that are associated with this network. And so this is a diagram that I put together some time ago, just looking at the sort of the architecture of our world. You obviously have the data and the specimens in the middle that are then associated with databases at all of these individual institutions in a very distributed format. These are then published out to aggregators, and the data is reused from those aggregators and repositories, things like GenBank, etc., is reused by this ever-growing external user community that is using data from natural history collections. Obviously, the primary mechanism is for taxonomic and systematic research, but there is an ever-growing list of people who are starting to use our data for all sorts of different end users that we sometimes may be unaware of, because all of this data is free and open to the outside world. And so being able to link all of this data together naturally requires everybody that's in this sort of complicated landscape to adopt this system and use it in order for us to be able to link all of this data together effectively and showcase all of those relationships between all of these pieces of data. So the focus of the Extended Specimen Network is obviously transforming our wealth of physical specimens and related digital data into an extended specimen network that will represent biodiversity held in not only the US biodiversity collections, but worldwide biodiversity collections as well. So as I mentioned previously, the Extended Specimen Network concept is more than just the digital specimens. The Extended Specimen Network concept includes 5 main focus areas that we have started looking at. One is collecting, actually going out and collecting more specimens and building our collections so that we can not only fill gaps, but can also do some long-term monitoring with our collections. And this involves people going out and not just collecting their own target taxa, but collecting more holistically, collecting more than just the specimens that they're interested in, but collecting on a sort of ecosystem kind of model. Also collecting all of the different digital concepts that you have associated with a specimen. Not only collecting the specimen, but collecting tissues, collecting parasites, collecting all of those linked pieces of information that are associated with that particular specimen. And then even including observation data, so things that you may see but not collect. Then obviously we need to continue with our digitization efforts. It's estimated that we've only digitized, a maximum of 30% of our collections. So we obviously need to continue that push of digitizing our collections and improving the digital footprint of our collections by providing more of that data online. Focusing on hard to digitize collections like entomology and paleontology collections, and also ensuring the completion of skeleton records that have been created by various disciplines, and improving the metadata associated with all of these digitized objects that we are then putting up online. Then we need to obviously look at the integration and attribution. We need to integrate all this data together at multiple different levels. Integrating at the preparation level, so associating all of these pieces of specimens together, but also integrating at the specimen level, so linking specimens together, like duplicates and hosts and parasites and those kinds of things. And then linking individual collection types together to be able to bring out relationships like tissue voucher, host parasite, plant pollinator, those kinds of relationships. And then integrating across disciplines. To bring data sets from outside our discipline into the realm of possibility. Things like ecology, geology, satellite data, geography data, those kinds of things. One of the other big concepts of the extended specimen network is the whole infrastructure component. Not only physical infrastructure to support the physical collections and the physical specimens, but also cyber infrastructure to be able to support the wealth of information that we are now collecting associated with our collections, coming up with some sort of community solution to data storage, and also the integration and attribution concepts that are required to link all of this data together. And then lastly, looking at education and workforce. Obviously, we need to train the next generation of cyber collection managers who are going to be looking after our collections. But we also need research initiatives, education initiatives to be able to bring our collections to the outside world and the various communities that use our collections for education. Some of the concepts of the Extended Specimen Network were highlighted in the recent National Academies of Sciences publication on biological collections ensuring the critical research and education for the 21st century. And you can see the link down at the bottom there if you want to go and download that publication and read it. So coming to my last slide, what comes next? We obviously need buy-in from all stakeholders. We need a global collaboration across all of these different players that are involved in this landscape in order to be able to bring about this concept of an open digital specimen or an extended specimen network that will be applicable to all collections around the world. So as Beacon, we have started some initial discussions with some of the players in this realm, bringing in people from the Research Data Alliance, GBIF, DISCO, ALA, IDIG Bio, and various others, to start talking about ways in which we can move this forward. One of the things that has come out of those initial discussions is the idea of a statement of intent, a statement that would indicate that all of these groups are interested in working together and working towards a common purpose. And Wouter will talk a little bit more about that statement of intent during the discussion period. We also then are hoping to work towards a Beacon-led series of workshops for further community engagement, looking at potentially discussing those 5 action areas of the Extended Specimen Network individually in a series of workshops to be able to flesh out what already exists, who is working on components of those particular aspects, and where the holes are, so where the gaps are that we still need to fill. And then hopefully that will lead into an implementation or an action plan for the Extended Specimen Network, where we can bring all of these people together. We can fund something both here in the US and internationally in order to be able to take this concept forward and make some serious headway in terms of integrating all of our data together into something that approximates an extended specimen network. And that is all that I have. So I would be happy to answer any questions.
Okay, so please put your questions into the chat and I will read them out for Andy.
Hello? Hello?
Okay.
Hello?
Hi,
May I continue? Hello, may I continue my question?
Sure, we can hear you.
Yes, can you hear me?
Yes.
Okay, thank you very much for your nice presentation. This is Tariku from Ethiopia. Hope that you know Ethiopia, yeah?
I do. I'm actually from South Africa, so I'm from the same continent as you.
Ah, that's great. I am very impressed about this concept, the extended specimen network concept. Fortunately, The Ethiopian Biodiversity Institute is working collaboratively with the SAMBI, South African National Biodiversity Institute, in spatial biodiversity planning assessments. That the project is funded by the CBD through the Japan Biodiversity Fund. So we are familiar with SAMBI, by the way. When I come to my specific question is that now we are on the way to implement the Green Global platform. Hope that you know that Green Global platform. That is the platform that used to manage the gene bank data. Mm-hmm. So to implement this well, we are working with the Crop Trust Fund. So now they are on the way to support us the technical capacity building areas. So we want to extend your concept, this extended specimen network concept, through capacity building approach, like long-term capacity building. It could be MSc or PhD program, because we want to catalyze that project through long-term capacity building to maintain the personnel. Because in the biodiversity and gene bank management, personnel succession is a problem. So once the personnel train, they may lose the institutes because of different incentive or other factors. But to maintain the personnel succession, we are thinking to devise a mechanism to maintain for a long time the personnel. One approach is we are thinking about the capacity building approach. So you mentioned that ECN is happy to welcome for research, and probably they will provide a fund for that. So in what modality can we work together in this regard? And then we can work together to share our platform data, say kind of things. Thank you so much for you listening.
Sure, yeah, I think that's important. You know, obviously there are a number of national and international initiatives that are ongoing. I know about the SANBI initiative. And so, you know, I think it is important for us to integrate this into those existing initiatives and try and support any of those sort of personnel issues that you have in terms of, you know, long-term support of personnel and making sure that you have a succession plan in place. And that is part of the extended specimen network concept, is looking after the workforce and the personnel. So yeah, definitely, it's definitely something that we need to work together on.
I've made a note of the intervention as one of the projects or initiatives to bring in, so we have a record of it. There are some questions from earlier in the chat, Andy. There was one from Carlos Martinez, which said that he had hoped to use Scratchpad's specimen content type to release specimen data online, but there was a malfunction that made that impossible. Which online platform can individual scientists and citizen scientists use for releasing data on digitized specimens?
That's an interesting one. That's outside of my realm of expertise, unfortunately.
I mean, I can comment on that a little bit from the DISCO perspective.
Sure.
Which is to say that during some of the survey and analysis work that the iStick project carried out during 2018, there were some specific tasks looking at what modalities, what mechanisms might be necessary to put in place to support scientists with private collections or groups of citizen scientists that were developing collections, and one of the conclusions was that you know, in the future that DISCO will provide services to to provide better support for that, but it isn't available today. There was another question from earlier in the chat from Deborah Paul. Let me just find it. She says, how can we address in the extended specimen data that much of the data across these different collection management systems is duplicated or replicated? And she gives people's names, taxon names, etc. she says so much labour, both machine and human, goes into keeping the very same data tidy as possible and filling the gaps over and over again. How can we figure out how to deal with this more effectively?
That is an interesting question. You know, unfortunately, it's a vagary of the way our system is built in terms of all of these distributed collections all around the world, sort of holding their own data. There are data cleanup initiatives that are ongoing through TDWG that have made some headway into this in terms of ensuring that all of these items are sort of deduplicated and shown to be the same thing. But how you would actually, you know, inform that process going forward, you know, I think there could be some better integration with those systems into collection management systems, so that information is getting into the collection management systems per se. There's also some benefit to having authority files generated for either, discipline specific or across the board, to be able to do things like people names, taxon names, those kinds of things. Obviously, a lot of the CMS systems are integrating taxonomic authority files already, and a lot of that stuff is being done by the aggregators as well. But it is a vagary of our system, the way that it exists at the moment, with all of these distributed collection management systems out there that are sort of looking after the individual collections themselves.
Okay, thank you, Andy. I don't see any further questions in the chat. Are there any?
I'm just going through them. I don't see any.
Here we are. There's one from Kari Lahti from Finland that has just appeared. How would or did you solve the requirement to be able to annotate the extended specimen data while the identical copies of the digital specimens are available for annotations in a number of repositories or web services with open access and annotation tools? I think what Carrie is saying here is that when you annotate one particular copy of a digital or extended specimen, how does that propagate?
Well, at the moment it doesn't. At the moment it's a purely manual process. So if anybody goes in and annotates a specimen through one of the aggregators, that is it's a total manual process of getting that information back to the collection so that the original record can be annotated. But obviously that's where, something like a blockchain inspired network would be able to help with that, where you would be able to go in and the annotation would, automatically be associated with the original record as a transaction. And you would be able to visualize all of that in space. As people go in and annotate or add additional pieces of information to a particular record, that stuff would be completely transparent to everybody who was in the network, and you would be able to link data, you'd be able to annotate data, you'd be able to do all of those things in a very sort of transparent manner.
Sorry, Andrew, can you drop your email, please?
I just said, yeah, it's in the chat.
I put it in too. Thank you, Tariku.
There twice for emphasis.
Okay, so if there are no more questions on the extended specimen presentation, let's go back to the agenda. Let's stop Andrew's sharing and come back to me. Okay, oh, sorry, wrong one. Okay, so now we're going to open up for the remainder of the session for a wider discussion, a free-form exchange of views and opinions based upon what you've heard in the introductions to digital specimens and extended specimens from myself and Andy. And the idea of this discussion is to try and identify the main similarities, but more importantly, the differences between the two concepts where we might want to think about convergence. Also other initiatives to bring in, as for example, the initiative in Ethiopia mentioned by Tariku, development and implementation plans, the potential role of the Research Data Alliance in conjunction with TDWG, and the next steps towards global convergence, including the statement or letter of intent that you heard Andrew mention in his presentation. So the floor is open, and we ask you to please raise your hand in the participants list, and we will try to keep track of the hands going up in the correct order. It will be a little difficult because the participants list is 100 persons or so. So please forgive us if we don't come to you immediately. We'll do our best.
So Alex, there's an interesting question in the chat that may sort of get the conversation started. I don't know whether we want to get involved in this too much, but there's a question from Ian Engelbrecht who says, has there been any development on the GWID front? It seems we can't agree as a community on what a GWID is. Alex mentioned PIDs, but will these be truly global or disco GWIDs?
I can answer that to some extent, at least from the disco perspective, and say that one of the important avenues that we have been investigating in disco over the last 12 to 18 months is precisely this question of how you persistently and unambiguously identify digital specimens. And we've studied and are still studying a number of options. And there are two key requirements, which is that DISCO itself has a requirement for a stable and robust persistent identifier scheme, and attached to DISCO's own requirement is the requirement that potentially that scheme should be expandable to the global level, so that all initiatives around the world dealing with digital or extended specimens can ultimately use a common scheme, because what we don't want to see is a fragmentation into different methods of identifying and dereferencing specimens. What I can say from the DISCO perspective is that we have settled on using some variant of the handle system. As many of you will know, the handle, one important incarnation of the handle system is the DOI system, which is used by DOI.org, the DOI Foundation, for the identification of journal articles. And that's not all that DOIs are used for. But we are still coming, although we have some preliminary conclusions on the way that we want to go. We're still in the final weeks of bringing that to a more determined conclusion, and we hope to be able to say something about it before the end of the year.
So one of the other interesting initiatives that has come to light recently is that the iSamples initiative has recently been funded to extend the IGSN system for geological samples into biological collections. So the IGSN system is a registry of identifiers for the geological group, and the grant that's just been given by NSF is to extend that out to biological collections. So they would act as a sort of central repository for global unique identifiers associated with objects, and thereby hopefully be able to bring about uniqueness of those identifiers, but also some sort of integration of those identifiers as well. And so they've been brought into the conversation fairly early to start looking at if there's a way that that this can be used as a system to be able to register and identify these identifiers to be able to link stuff together.
I think some information has just appeared in the chat about that, yes, from Sarah Ramdeen, who represents the IGSN initiative. I don't see any hands up in the participants list at the moment. But if somebody wants to dive in and make a remark, please feel free to do so.
We've stunned them into silence.
We have. Oh, Demetrius has his hand up. Please go ahead, Demetrius.
I promised myself not to speak today, but just to make a start. So what impresses me, and I would like to highlight this, is listening to you, Alex, and listening to Andy. What is very important that we all understand, I think, from the two presentations is that the two lines of thinking, although obviously very connected, they came to this more or less the same conclusion quite independently. And I think that's very important, because it really shows the maturity of the community and the need today to embark on these kind of discussions and also look into the implementation mechanisms. And I think this is one of the rare situations within the community where we have this kind of a consensus from the point of view that is not really driven from funding. We have not been funded from the same source. We actually have not even fully communicated these developing concepts for some time until we did. And listening to the two presentations, I could rarely distinguish between the two concepts, at least in the sense of the scientific benefit that would provide to the community. Of course, there are many discussions about the implementation, and this is why we're having these discussions here. But I think I wanted to highlight this, and I think this is one of the positive notes that is important for our community to highlight.
Yeah, thanks, Demetrius.
Yeah,
that is really true. You know, and one of the early discussions that we've been having in the discussion sessions that we've been having through Beacon is how would we fund something like this on an international basis? You know, obviously there are initiatives going on in the US that are funded by NSF. There are initiatives going on in the EU through DISCO. There's initiatives going on in Australia and Australasia through ALA. There's initiatives going on in Africa through SANVI and various others. So how do we, you know, converge all of this together, and how do we fund it as a sort of global mechanism that everybody can get on board with? You know, obviously there needs to be some component of countries or regions not only funding their own local initiatives, but also pumping some money into a sort of global infrastructure to bring everybody together and drive everything towards a common purpose. And I think that's an important part of ensuring that, you know, everybody stays on the same page, and we're all integrated together in our thinking and moving this forward.
Okay, so there's a comment and a question in the chat from Tim Robertson at GBIF, and he says some things that I think many of us know already, which is that it's GBIF's impression that many collections are managed in databases like FileMaker, Excel, etc., and indeed surveys have shown that there's a very wide range of different collection management systems in use. And his question goes on to say, what would you recommend that the community do now to prepare for the forthcoming OpenDS and extended specimen network work? And I think my answer to that would be get involved. I think this isn't a short-term piece of work. It's not going to be all done and dusted in just a few weeks or a few months. It's going to take a year or two to do, I think. And what we really need is participation from different parts of the community that are expert in the different ways that information systems, collection management systems, etc., are used within institutions and elsewhere, to help think about what is the sensible path from the situation we have today to the desirable future that we would like to see in some years' time. So becoming engaged and help planning and defining that work and sending it in the right direction would be really very helpful first steps from the community.
Yeah, I agree. I'd also like to believe that dynamic is changing. that collections are starting to realize that, you know, in order to be part of the global community, you have to really have a true collection management system that is managing the digital data associated with your collection, and that, you know, things like FileMaker and Excel just don't cut it anymore. You know, and so hopefully that dynamic is changing, and more collections are moving across to, you know, to a fully fledged collection management system that can not only manage their digital data, but can also publish it effectively out to the outside world to make those linkages.
But I think that there are issues of capacity and competency here as well. You know, we all know that, you know, collection holding institutions vary greatly in their competencies and their capacities to deal with collections management using information technology. And you know, this is certainly a problem that DISCO recognises in across the, you know, the 120 or so museums that are participating in DISCO in Europe. And again, I think, you know, some of the elements that DISCO is putting in place to deal with match very much some of the things that you've been saying, Andrew, in the ESN initiative to do with infrastructure and capacity building. You know, we've identified that, you know, that DISCO needs to provide training and support services to improve capacity and competencies. But also for those institutions that don't wish to manage their own collection management system installations, to provide collection management systems as a service in the same way that, you know, other industry sectors provide software as a service to their clients and customers. And I think that you know, there are real opportunities for some of the existing, you know, sustained and supported collection management systems to become candidates for CMS as a service, operated by, you know, infrastructures like DISCO or iDigBio or elsewhere. Yeah,
I totally agree.
There's lots of new messages appearing in the chat. Ian Engelbrecht says SAIAB. I don't know who SAIAB is, I'm afraid. South Africa.
Biodiversity.
Yeah, has been doing that with Specify.
Yep.
So already we have at least one example of CMS as a service.
Yeah, Specify has recently started offering Specify 7 in the cloud as a service, and, you know, offering a lot of the capacity building initiatives to facilitate these, you know, smaller collections who don't necessarily have the infrastructure to house their own servers and their own databasing systems, you know, for Specify to be able to do it for them.
Yeah. I mean, certainly DISCO has you know, commenced discussions with both Specify and the DINA consortium as well, you know, in relation to the digital specimen and extended specimen concepts. But of course, there are many other initiatives as well that need to be engaged.
So there was one a little further back from David Shorthouse. He says, "What is the single most important carrot? In other words, services or metrics for data publishers that would convince them that there is no other viable option to share specimen data except as digital extended specimens?" I would say probably the single most important carrot is the research that can be done on those specimens. You know, our ability to be able to integrate all of these pieces together really improves the kinds of uses that these specimens can be put to. You know, in terms of being able to answer the big questions like climate change, invasive species, you know, those kinds of things. You know, I think unless your collection and the specimens that it holds are part of that digital infrastructure, you know, those the specimen data associated with them, it's very difficult to use it.
At least in Europe, I don't know how it is in other continents, but at least in Europe, it comes down to being able to associate, you know, societal or economic impact that is derived from, you know, work that is funded out of the public purse. So the easier it becomes to demonstrate the impact of specimen collections, the more likely it is that such collections can be sustained and enhanced and improved.
Demetrius, is your hand still up from previously, or did you want to make another comment?
No, I was aiming for another comment, but...
okay, go ahead.
Yeah. No, I'm reading through the questions and the comments in the chat and listening to you guys talking about this. I get this feeling that many people think, okay, that's a good idea, all right, it's a convincing way forward and it makes probably sense. But how do we get the critical mass of organizations to not only buy in, but implement and mature in a way that are able to become more open, digital, extended specimen ready in the future, whatever we want to call it. And I think this is when an interesting discussion comes in on how we gradually see the institutions either at national level or at the regional level, and hopefully in the future at a global level, start to think about their joint capabilities, start to think about solutions that go above and beyond their institutional future and see their common future, either through the aggregation of the collections at distant level or through policies and practices, and how they can actually leverage resources and funding at the regional or international level to do exactly that. So I think the question about the sustainability of these systems needs to go through also the kind of new model of collaboration that we see emerging definitely within Europe, i.e. DISCO, but I would say also now in the US and in other places. I mean, we had these discussions also in Australia recently. So I think, you know, this all goes hand in hand, and we cannot talk about the future implementation of this kind of solutions without talking the required change in the paradigm through which we collaborate. And I think we already see this kind of very positive signs. I think this needs to go hand in hand, definitely.
So there's a question from Abby in the chat. She says, do you have a recommendation for how to keep data linked to the specimens in the interim while we continue to work to get this system in place? For instance, I know of a researcher that has trace element data from marine mammal specimens, but the system is not equipped to handle that data. How do we keep them linked until we can get something like the extended specimen concept in place? So I know one of the ways that I've been tackling this in my collection is literally through a manual process of going in and attaching GenBank sequences, publications, CT scans, all of those kinds of things to the physical specimens in my collection, and then publishing that data out to all of the aggregators. So if you go to one of my specimens in GBIF or IDIG-Bio, you'll see that there are, you know, GenBank sequences associated with those individual tissues. There are all of the original publications that those GenBank sequences were published in, CT scans that are sitting in Morphosource. All of those linkages are being made in a manual way. I mean, it's hundreds and hundreds of hours of my work in order to be able to go and do that. I have, you know, over 17,000 GenBank sequences that I've been able to scavenge from GenBank and through the literature, and connect those up to the specimens in my collection. But it is very much a manual process, and it's labor intensive. You know, and that's the only way that I can think of to, you know, to make those connections until we have, you know, a technological solution that will be able to link all of these things together automatically without me having to go through the manual labor of getting it done.
Okay, there are a couple of quite specific questions in the chat. There's one from Brenda Nyaboki in Kenya, specifically about which collection management systems to use, and there's one from Elizabeth Womack about formats of observation data. I propose that we will treat those offline and get back to you with answers for those, if that's okay.
Sure, Brenda, if you want to get in touch with me, I can certainly give you some information about Specify.
Yeah. I mean, coming back to this question about you know, how can we get things moving? There was another comment in the chat. I can't find it now. It was Deb Paul made it in response to something Eli Wallace said about getting software engineers to work more closely together. And I think that you know, developing a common specification is one good way of doing that, because generally speaking, the best specifications, the best standards come as a result of implementation lessons, good and bad, that engineers make while they're trying to implement some of this stuff. And so we would be looking for the engagement of software developers in the specification process.
So Alex, a question for you. Do you see any similarities between your sort of implementation of OpenDS and a sort of blockchain kind of inspired network concept of a sort of transactional system of being able to, you know, being able to visualize all of these transactions that are going on a particular specimen at any one time, and thereby not only being able to track the integration of data associated with specimens, but also be able to track things like loans and movement of material around the world that is necessary for things like Nagoya and various other, you know, initiatives that are ongoing.
Indeed, I think there's a whole class of things that you do to specimens that can be described as transactions, and you know, you talked about them in your talk, and I did to some extent as well. You know, arranging loans and visits of the physical specimens. You want to know, you know, who is visiting it and who is borrowing it. You want to know where the physical specimen is at any particular point in time. Much of that is about asset management, rather, but some of it has a scientific angle to it as well, because you want to know who is using the best specimens. Annotations and interpretations, etc., can also be, you know, I suppose, lumped into that category of transactions around a specimen. And indeed, you could probably consider that you know, updating the data or adding new data about a specimen or derived from a specimen is another kind of transaction for which you want to keep a change history or a provenance event. And so, yes, I think the transactional approach to dealing with a digital or extended specimen can be very helpful. I think the question of whether you implement that using a blockchain or not should be considered as a completely separate question, because you know, there are many different ways that you can implement you know, transactional record keeping. Historically, it's done using relational databases, of course, in banks and that kind of thing. But many of us will be aware of the emergence of blockchain and its association with cryptocurrencies. I have to say that blockchains are not just for cryptocurrencies, and that's again, that's something that should be separated, because they have other uses too. One of the things that's extremely interesting about a blockchain is that from the point of view of principle, every block in a blockchain is in fact a digital object with a persistent identifier. And so it makes the blockchain concept on one level extremely compatible with this idea of digital specimens as you know, representations of physical specimens on the internet. Beyond that, DISCO hasn't. I mean, it's done some thinking, but it's not yet done any experimentation in that area.
Because the problem that I see with a transactional method like that is that it would require a ground shift in the way that we currently publish our data. You know, currently we publish our data as sort of, you know, time-stamped snapshots. And in order to be able to, you know, publish that whole transactional system, it would really take a ground shift in the way that we actually publish our data out to the outside world.
Okay, so shall we move on to see if we can identify what some of the next steps towards better convergence, or at least avoiding fragmentation might be? I mean, I think Andy, you talked about the fact that the Biological Collections Network has been thinking about this and taking steps in that direction with thinking about acquiring funding to run some inclusive workshops. And you also mentioned the letter or statement of intent, which I think it would be appropriate to ask Wouter to elaborate on a little more maybe at this moment.
Sure, sounds good.
Wouter, is that appropriate for you to do right now?
Yes, just looking up the link to that, so I put it in the chat. Yeah.
Do you want to share your screen as well with it?
Could do that. Let me see. Share screen.
I can bring it up if it's easier.
Can you see this?
Yep.
Yes.
Okay.
It's a little small, but we can see it.
Okay. So yeah, the idea behind this is I think it's twofold. First of all, we have had these initial concepts being raised both in the US, by Beacon, the extended specimen concepts, and the digital specimen concept in DISCO. And as I said in the chat, yeah, that basically is a very good thing, because it shows that in different parts of the world we have this very similar needs. But for many people, it's not immediately clear that we are actually talking with each other, and that we are discussing what we are doing now in this session, how we can align our efforts and align these concepts maybe towards one global specification or multiple specifications that are interoperable. If our use cases are slightly different. So we have been talking with a few partners like Beacon and DISCO and GBIF and a few others. It would be good to have a letter of intent that shows that we are collaborating on this. Also, this letter can show, if it's signed by many organizations, that there is a wide global support for this. And as Andy mentioned, that would be kind of mandatory to make this concept successful. So this letter is quite short. It expresses 6 different statements that the signatories agree, and this is open for signing, both by organizations, but also individuals that are participating in this meeting could sign this if they agree with these 6 lines. First of all, there's the recognition of the need of an enriched specimen information model to link derived or related data back to the specimens. There's the recognition of the need for interoperability to enable local, regional, and global aggregation of specimen data as the anchor point to link all the other data to, in addition to traditional anchor points like species names or occurrences. And then the intent to collaborate on converging these concepts that have been discussed here today. The aim to collaborate in a global process that's open to participation for all stakeholders under the umbrella of the Alliance, and also with stakeholders from the Earth sample domain, because the specimens are not only biological specimens, but we have also Earth sample specimens in our collections. To strive to develop different modes of collaboration, and to identify regional funding opportunities. As Andy already mentioned, we can also look at NSF funded research coordination networks, for instance, and in the European Union there are cost actions that could be used for this, etc. And then that we together seek to the most effective strategies to develop biodiversity knowledge capacity in all countries of the world to ensure that they benefit from collective biodiversity knowledge, both within their own countries and beyond. Okay. So everybody who agrees with this, both organizations and individuals could sign this, and we intend to use this in future communications to show the world that we are discussing to work together on this and to align our efforts.
Okay, thank you, Wouter. I think that's very helpful. I think another dimension to this is, if you recall, this is a birds of a feather session where, as a group of interested people in the, under the TDWG umbrella, we're exploring the question of digital and extended specimens and convergence. And I think there are, it's right that we think about what role can TDWG play in this in the future, and also what role can one or more of the interest groups within the Research Data Alliance, and I'm thinking specifically of the Biodiversity Integration Interest Group and the Data Fabric Interest Group can play in this. I wondered whether any representatives of the TDWG executive or the co-chairs of one of those RDA groups had any thoughts on the matter.
Well, I guess I can say from the TDWG perspective that we are very open and welcoming of this letter of intent. We are, as I said in those of you who came to the introduction sessions for TDWG, the Biodiversity Alliance is something that we believe strongly in, these services that must be sustained for our community. And so certainly TDWG is a key player here and as we were showing here, sort of a key forum for discussing all of these pieces of the puzzle that at least are within our domain. And we look forward to contributing as an entity however we can.
Okay, thank you, James. I don't think that we've got any of the co-chairs of the RDA's Data Fabric interest group here, but as a participant in that interest group, I can probably offer the view that interest group would be certainly willing to support and see the development of this further work along the lines outlined in RDA recommendations.
Joe, I see your hand up. Go ahead.
Thank you. I just want to explain a little bit about the Alliance for Biodiversity Knowledge. So this is probably a little bit less well known in the US than it is in Europe. But GBIC2 was a meeting that was held 2 or 3 years ago in Copenhagen, hosted by GBIF, to develop the community to answer and work towards solutions for these very type of questions. And out of the GBIC2 conference came the Alliance for Biodiversity Knowledge, and the goal of the Alliance is to facilitate these type of conversations to develop products and solutions to global biodiversity needs. So I'm very happy to represent the Alliance in saying that this is exactly what the Alliance, that the community needs, and that this is a great move forward for the biodiversity, this community. And I'm really happy that it's happened organically. And I think it has a lot of strength to it. And if you need, I'll put a link to the Alliance in the chat. So if you need more information, but you'll definitely hear more about this under the umbrella of the Alliance as well.
Okay, thank you, Joe.
Thanks, Joe.
Alex, I have a comment, if it's okay.
Sure.
Yeah, that's Gil. Is Gil, is it? Yes, go ahead, please.
So, so I have signed this letter of intent on behalf of IDIC-Bio. We are very interested in this. We've been working very closely with ALA, GBEF, DISCO, others for a good long while, and we've been working on this same idea, same issue, and it seems to have, as Joe mentioned, organically just blown up and become a very important and a very well coordinated activity. And so we are certainly supportive and intend to work closely with the Alliance and with RDA to and Beacon and all the other folks represented here to pull this off.
Okay, thank you. Thank you, Gil. I note also an important comment that that William Elliott has made in the chat. He says, since we're talking about global systems, resources and funding, let's not leave developing countries out by providing services that also consider their needs from the start and to integrate them as a way to reduce the digital gap. Yeah, that's, of course, yes, I support that view too, as do others in the chat.
Yep.
Deb says 100 people support it, so. Thanks, Deb.
I think she's just voting 100 times.
Yeah, okay. So Lauren says, any thoughts on how we might do that?
Well, I think involving a number of these initiatives, like, you know, the SANBI initiative in South Africa that was mentioned, you know, involving some of these initiatives in the early process of discussion would be a very important mechanism for ensuring that that happens.
I would certainly, from the European perspective, I can report that over a period of 6 years or so, we did closely involve SANBI in some of the coordination work that we did, particularly around the preparation for the LifeWatch research infrastructure. But there has more recently there has been, I think, I don't know that it's a change of direction, but certainly a change of personnel within SANBI, which means that they've become less engaged than they were previously.
Is anybody aware of similar initiatives ongoing in South America or Asia or?
Not specifically in South America or Asia. I'm aware of some initiatives in Australia in CSIRO, but I've not had any contact there for some months.
We have pretty regular contact with the CSIRO folks. So if the question is, how do we move all of this forward? Then I think what Andy outlined in his presentation regarding a series of workshops, and I mean, I would have to take this back to the IDIC-Bio steering committee, but as soon as we're free to, I think we can start this virtually, but as soon as we're free to do things in person, then I think that IDIC-Bio would support Beacon and some of the work there with global workshops so that we leave no one out and we invite these countries that might have small collections or might have resource problems to see if we can't bring folks together to begin working on this under the umbrella of the Alliance. So I think there's probably enough people in this room with enough resources to figure out how to pull people together. I don't think we should leave anyone out in what we're trying to do here.
No, I think that's exactly right, Gil. And as you began to say those remarks, you know, David Shorthouse also made a suggestion in the chat about, you know, both or several teams to build the most basic Hello World implementation possible. and leading towards some public demonstrations. And of course, there are some learning experiences there that then feed into the specification work. And certainly, it's the case in Disco in Europe at the moment that we have embarked upon some prototype development work. We did quite a lot of development work during 2019. It's a little bit stalled at the moment because of lack of resources. And we've been supporting discussions in the US as well about trying to kickstart some of that kind of development work there as well.
Joe, I see your hand up again. Go ahead.
Yeah, thanks. So earlier this year, Tim and Donald ran a workshop that was a virtual webinar on the collections catalog, in which I guess that was kind of funded out of the Synthesis Plus grant. However, we actually developed that as a virtual webinar before COVID. So, but it came out to be good timing. So and with that, a white paper was written to kind of guide the discussions of where this collection catalog discussion program should go in the various portions of it. So a way forward would be able perhaps to build that discussion around a white paper or the many different threads that we have here, put them in a structured format and have individual discussions around that, and then guide these potential workshops around them, because we are funded in the Europe with Synthesis with 2 more workshops, and then there are other opportunities as well, and perhaps divvy out the workshops in the timeline between the European funded projects and the US and hopefully other partners as well. I certainly think that would be a good way forward, especially with all this momentum that we have here today.
Okay, thank you for that suggestion, Joe. I think it's a very helpful one.
Yeah, and I think that online mechanism that you guys used to guide that discussion was a very interesting one. I think it was very successful. So I think we can certainly implement something similar for this. You know, to try and engage more people in the discussion and try and get them all involved.
Yeah. It's a lot of work, and that was kind of a hesitation, but with the interest that we have here today, I'm not doubtful at all that the energy is there to do it.
No, I think the energy is there to do it. You're right. I know that Tim has said the final report from that piece of work is not yet written, but I'm sure it's in process and won't be too many more weeks before it's available. And very many people will be looking forward to that, of course. But I think it's a good suggestion, and it's one that we should take away and plan how we can tie it in with what Disco is envisaging in terms of next steps, outlining the design of OpenDS more formally than it is at present, and what Beacon is thinking about in terms of next steps with funded workshops. Okay, so I don't know what you think, Andrew, but I'm beginning to feel that we're coming to a natural end or conclusion to this extremely helpful birds of a feather session, and that we can probably begin to wrap it up.
Yep, I would agree. Thanks everybody for your comments.
Are there any final comments or remarks that anybody wishes to make?
We will certainly be trying to publish the availability of these workshops once we get them all set up as widely as possible to try and get as much engagement as possible. So look out for those announcements coming out through your usual streams, and hopefully we can get everybody involved and as much of the global community as possible.
I've just taken a look in the Google Doc notes document, and I see that Walter and Gil have done an excellent job of capturing much of this discussion. So thank you very much to them for doing that. I have saved the chat, so I will paste the chat into the end of the notes as well, and we will tidy it up a bit so that and make it publicly available. I will remind you about the letter of intent that Wouter introduced, and encourage organisations and individuals to consider signing it. It's available immediately for signing at the link that Wouter has just repasted into the chat. Oh no, or is about to repaste into the chat. He's just put the notes there, but I think he'll repaste the letter of intent as well. So it just remains for me to say, I guess jointly with Andy, thank you to everybody for your contributions. It's been extremely helpful and useful. I see that there's a lot of enthusiasm and support for these ideas. And even if not everything is fully understood or agreed, there seems to be no fundamental dissent from everything I've seen and heard. So thank you all for participation, and thank you again to the various helpers, Gil, Wouter, Matthias in the background, members of the TDWG executive for organising these working sessions. And Andrew, I want to say thank you to you also for co-organising this with me. I've valued very much the input from the Biological Collections Network and the US side. It helps me personally fill out the picture I have of what's going on in the world. Thank you.
Yeah, thanks, Alex. Back at you. It's been good to collaborate and try and come to a common goal. So
yeah. So I guess we'll have a debrief sometime after this.
Yeah, sounds good.
All right. Okay. Thank you, everybody. Thank you very much. And let's say goodbye to everybody now. Bye-bye.
Thanks, everybody.